Good afternoon, Hoylake and West Kirby. This is Imaging Atoms, Molecules and Cells. I'll be giving you an introduction to where I started in the world of imaging by looking at atoms and then slowly working my way up to molecules. I'll be taking a little bit of a detour to explain why it is that when we look at images, not necessarily images of atoms and molecules, but images more generally, why analysing images can be such a challenge, because human perception of what we're looking at is not necessarily the same as what it actually in an image. And I'll be looking at spin-offs of what I've been doing over the last few decades in terms of looking at images, spin-offs in the earth sciences and in the medical sciences. And I'll spend a lot of time at the end looking at what I've been doing most recently, which is looking at complex systems. And complex for me means not something as simple as an atom, but complex means, for instance, biological systems, which are full of thousands of different types of molecules. And I'll finish with looking at what I'm doing at the moment, which is looking at spectroscopic and imaging studies of cancer. So let's just make sure we've got the basic concepts right. When we're looking at images in a scientific context, two things are important. When I talk about image processing, I mean about taking an image and doing something to it, processing it, changing it in some way in order to help us, meaning humans, interpret it. In other words, we can take an image and we can, for instance, do something very simple with it, like adjust the brightness or adjust the contrast or adjust the colours in order to make the information content easier for us to interpret. Separate from that is image analysis, which means taking an image and extracting some of the information that's in that image, quantifying it. In other words, converting an image into a set of numbers. What those numbers are, well, it depends on the context of what you're trying to get out of those images. So image processing and image analysis is a lot of what I've been doing in my research for many decades. I started image processing and image analysis way back in the 1980s and early 1990s, and although my research interests have changed over the years, this aspect of research has been with me for quite a long time. So I'm going to be illustrating this talk with images taken from research projects that I've been involved with over the years, some old, some new, and some from collaborators as well as students that I've supervised. This is an image taken from a scanning tunneling microscope. It's effectively the microscope that gives you the highest magnification. Notice that this particular image has got a scale of less than 1 nanometer. So 850 picometers, 1 picometer is 1,000th of a nanometer. There are 1,000 nanometers in a micron and 1,000 microns in a millimeter. So you can see that this is a very high magnification image. And that's why we interpret these little blobs there when we're looking at the surface, in this case of carbon. We would interpret those blobs as being individual atoms of carbon. And just to give you a sense of scale, on this particular magnification, at this scale, a grain of sand would be about the size of the Earth's moon. Gives you an idea of the magnification we're dealing with. So whenever I look at an image like this, I always tend to think of William Blake's poem, Starting to See a World in a Grain of Sand. So the device that produced that image is the scanning tunneling microscope. It's in the stainless steel chamber behind the handsome scientist there. It's in one of the research centres in the University of Liverpool. It's encased in a vacuum chamber in order to keep the system ultra clean. And the principle of a scanning tunneling microscope is not using light to take images like an optical microscope would, but a sharp metal tip is moved over the surface. Here we're asking what would it look like if we were to zoom in and see individual atoms. Well, a sharp metal tip might end in one or a small number of atoms, and if we're looking at a surface which is not necessarily atomically flat, then representing the atoms as these little spheres, we can see that there are various bumps on the surface, and so the distance between the tip that we're moving and the surface will change as we move and take the tip backwards and forwards. And this small change in distance, even though it might only be measured in a fraction of an atom, those small differences in distance are enough to actually measure a current that flows between the tip and the surface, and that allows us to map out what the topography of the surface looks like. So this is an image taken by a colleague in the University of Manchester, and here the individual bumps that we see on the surface, the scale here has been exaggerated for clarity, but these bumps represent individual atoms on the surface, in this case the surface of rust. This is an iron oxide. And for those of you who remember your chemistry, iron oxide can exist in various phases, Fe2O3, Fe3O4. And what we're looking at here are different phases of iron, which is why we see these small islands, which appear to have individual atoms. And then between these islands, there seems to be a rather smoother region. We interpret this as being different phases of iron in which the atoms show up with different contrast, and in this case a different apparent height above the surface. Note here again the scale, that is 2 nanometers on the scale bar, and that's why we think these individual bumps are individual atoms. We've had the technology to see the world on the atomic scale for quite a few decades now. This technology was invented back in the 80s and refined in subsequent decades. Working with scanning tunneling microscopes, or STMs for short, led to the development of a particular type of analysis software that I developed for my students. Because this idea of scanning microscopy can be applied in lots of different contexts, some of which you might have heard of and some of which you probably haven't. So for instance, over the last few decades, different microscopes have been developed, such as scanning Auger microscopes. You probably have heard of scanning electron microscopes, but there's also scanning force microscopes, scanning ion microscopes, scanning laser microscopes. There's all sorts of scanning microscopy. Scanning optical, scanning tunneling, I've just mentioned, that's what we've just been looking at. So there are lots of scanning microscopy where there's a word in the middle that describes how the images are actually acquired. And you can refer to all of these types of scanning microscopy as SXM, where the X refers to either tunneling or Auger or optical or laser or lots of different acronyms. And that's why when I wrote software to help analyze these images, I called it image SXM. Unpronounceable, but it describes the fact that it's designed for scanning microscopy where the X refers to lots of different techniques. I wrote this software so that my PhD students would have software that allowed them to look at images from lots of different types of microscope. Not just different types of microscope, but perhaps the similar microscopes from different manufacturers, all of whom have their own way of storing the data in the image files. So that's why for many a decade I was involved in writing and designing and updating and extending software that allowed us to process images and also analyze images that were coming off these microscopes. The software I wrote has been used by researchers in various countries around the world. So here there's a pin for every country for whom I think there's a user somewhere in there. You can see I'm having a problem with Greenland, and I've never really been able to break into Antarctica. But so far, it's nice to know that the software which I wrote for my own students is now being used by thousands of researchers throughout the world. I used it primarily, and my students used it a lot, for looking at individual atoms on surfaces, looking for the arrangement of atoms. I was particularly interested, back in the 1980s and 1990s, I was particularly interested in those elements called rare earths, the ones at the bottom of the periodic table. I was interested in how the atoms arranged themselves and how that affected their properties, their material properties, their mechanical properties, their magnetic properties, their chemical properties. How were all of these things affected by the arrangement of atoms? And in a given crystal structure, if you look at the crystal structure from different directions, or you look at how these metals can be grown on top of other transition metals, that gives you different structures, all of which give you different geometric arrangements. If you look at a scanning tunneling image of a particular rare earth, this one happens to be scandium, we see that there are terraces. A few white blobs indicates there's a little bit of contamination on this surface, and these terraces tell us something about the way the atoms are arranged. And for quite a few years I was interested in these rare earth metals, and that led to the writing of a book on the surfaces of rare earths. I co-wrote that with one of my postdocs. It's interesting to see what you can do with image processing and image analysis. This is a surface of a particular metal alloy taken by one of my colleagues at Liverpool. He doesn't like me using this because it's a rather poor quality image, but I like it because it is a poor quality image, and it's a very nice example of how you can improve an image by using image processing. So here we would like to know the arrangement of all of these atoms that you can see in this mottled background. Not the large white blobs. The large white blobs are just probably individual atoms of oxygen which have found their way onto the surface, so they are just contamination. We're interested in the arrangement of all of the rather grayer blobs in this mottled background. It's difficult for us humans to see what's going on. So how can we process this image to make it clearer? Well, we have reason to believe that the atoms on this surface are probably arranged in a particular pattern with either 5-fold or 10-fold symmetry. In other words, if you take the surface and rotate it by 1/5 of a revolution or 1/10 of a revolution, the atoms will tend to look very similar. So we have reason to believe that there is a symmetry involved. And we can use a technique called Fourier analysis that tells us what particular frequencies are involved, what particular rotations might be possible on this surface to make the atoms look the same arrangement. And if we use this so-called Fourier analysis, we get a whole load of dots, which are at first sight a little difficult to interpret, but if I highlight some of them, you realize that they are in rings of 10. Indicating that yes indeed the atoms on this surface are arranged in 10-fold patterns, even though our eyes find it very difficult to pick up that information from the left-hand image. Not only that, but we can use the fact that we have identified a 10-fold pattern in the data to actually reconstruct what would that surface look like if we only kept the 10-fold pattern. In other words, throw away everything else, throw away the oxygen atoms and just keep the 10-fold patterns. You might say, well, isn't this cheating? Aren't we just sort of forcing our prejudice onto the image? Are we not just guessing that these things are actually in the patterns indicated by this orange image here? For instance, if we have a look more closely at what's going on here in the blue circle, it looks like the atoms are arranged in a sort of sunflower, a 10-petaled sunflower. Is that just a consequence of the way we've done the image processing? Or are the atoms actually in those positions? Well, if we look more closely at the original image at exactly the same location, as shown by the red circle, well, we find that yes, there is a collection of 10-fold pattern there. There is a 10-fold sunflower petal there. It's just easier to see in the middle image than it is to see on the left-hand image. So yes, it looks like we have genuinely processed the image to make the detail easier for humans to see what's going on. And we've used mathematical formula and mathematical processing in order to do that. But we do have to be careful that whenever we process an image, we're genuinely bringing out what was already there rather than simply adding things and making artifacts. When it came to looking at molecules on surfaces. In this particular case, there's a molecule that will go onto the surface, that will bind to the surface and will react with the surface, and the molecule sits on the surface of this substrate. It doesn't really matter what the substrate is. And these molecules can arrange themselves in one of 4 orientations, as indicated by the panel on the left, and can be represented by the 4 symbols there, the 4 triangles. So I was interested in analysing the image and not necessarily processing it, but analysing it in order to pick out what are the orientations of this molecule on the surface. Is there a particular preference? Does the molecule want to sit preferentially in the orientation indicated by the blue triangles rather than the red triangles, for instance? So I analysed the image, used my software Image SXM to analyse the image and try and pick out what those orientations were. And then I generated a sort of false image indicating what the orientation of the molecules are and labelling them according to those triangles. And that makes it easier for a human to look at that and say, well, there appears to be a mix of blue and red. It looks like there's a few more blue than red, but there's no particular preference as to whether the triangle points up or the triangle points down. So although in principle that contains the same information as the original image of the molecules, it's now easier for humans to interpret it. We don't always have this problem, for instance, changing scale somewhat. If we were to photograph, in this case, the Milky Way, here's the Milky Way photograph from a very dark site, and therefore there's very little light pollution in order to contaminate the original image, and so it needs relatively little image processing to bring out the details that are there. But if we were to take a similar image, not of the Milky Way, but of a particular nebula, if we were to take an image under the light polluted skies that are quite typical in the United Kingdom, then we find it very difficult to see what's going on. So in this particular image, there is a big cloud of hydrogen in the middle there, but it's almost impossible to see because there's so much light pollution that's giving us a background that makes it difficult to see what's happening. In this particular case, we can improve what's going on by adding lots of images together. So here, Image SXM has stacked a whole load of images together, which then makes it easier to pick out the nebula, this object in the middle there. You can perhaps see why it's called the North America Nebula from a rough outline, something like the coast of California and the Gulf of Mexico, etc. And once we've got an image like that, well, we can do a little bit of tweaking and, if necessary, enhance the colours a little bit to try and bring out the detail. But again, this image processing is bringing out detail in the image which was already in the image. The detail, the information is there in the left-hand image. It's very difficult for humans to see it. By processing the data, we can then bring out that detail to make it easier for humans to see what is actually going on. But let me take a diversion at this point to say why it is that this is not necessarily a trivial task. Why is it that extracting information from images can be so difficult? It's the difference between human perception and reality. How we perceive images, in other words, what we see or what we think we see, can be very different from actually what is contained in an image. In most day-to-day situations, we just trust what we see and we don't worry about the fact that it's not necessarily what's actually in front of us. Scientifically, though, of course, we can't do that. We have to worry about whether human perception is being fooled by what it is we think we're seeing. We can ask ourselves, well, which is the better image processor? Is it our brain, which does a lot of processing of what it is we're looking at, or is it a computer? But bear in mind that, well, basically they're both computers. Brain is just a computer made of carbon, and what we normally call a computer is basically a computer made of silicon. Does it really matter what the atoms are that make up the computer? Well, in terms of our perception, it matters. And let me give you an example of how what you think you see is not necessarily what's actually there. I hope not too many of you are colorblind. If you are colorblind, I'm sorry, this particular demonstration will not work for you. Let's just have a think about what it is we're looking at here. We're looking at a cube, a 5 by 5 by 5 cube. On the left-hand side, I'm looking at a cube and I'm only interested in the colours of the tiles on the top face of this cube. So you can see 25 coloured tiles there, I hope. I'll give you a couple of seconds to count how many blue tiles are there on that top face on the left-hand side. Okay, you've got a number. Now have a look at the cube on the right-hand side of the screen. Again, it's the same sized cube. Now I'm interested in looking at those top 25 tiles and asking how many of those tiles are yellow. You should now have in your head two numbers, the number of blue tiles on the left and the number of yellow tiles on the right. But the point of this particular slide is to tell you that the blue tiles on the left, and I'm assuming that you've picked out these 4 tiles on the left-hand image, and these tiles on the right-hand image, I'm assuming you've picked out those 7 yellow tiles. The 4 on the left and the 7 on the right are actually the same colour. Yep, I'll say that again. The 4 tiles I've just pointed to on the left and the 7 tiles I've just pointed to on the right, the ones that you think are blue on the left and are yellow on the right, are not. They are actually the same colour. And let me show you that by taking away all of the pixels in this image other than the pixels in those 4 tiles on the left and 7 tiles on the right. And I'll do that one step at a time. And there they are. I've now removed all of the other pixels which were confusing you. Those are what you are actually looking at. The blue tiles aren't blue, they're actually grey. The yellow tiles aren't yellow, they're actually grey. And they are exactly the same shade of grey. I've checked them myself. If you don't believe me, you can go to the website in the bottom left there, lottolab.org, and convince for yourself that this is not my trick of PowerPoint. That is actually the way it is. In having lots of pixels around the ones I was pointing you towards simply confuses your perception. They're not blue. They are simply more blue than the yellow surrounding pixels. The ones on the right aren't yellow. They're just yellower than the surrounding blue pixels. And if we take away the confusion, then your eyes are in no doubt that these are all the same colour. That's how easy it is to fool your eyes. So if you think when you look at a coloured object you are actually seeing the colour of the object, well, maybe you are, but maybe you're not. And this idea of perception being easily fooled is not simply one that's confined to colour. Simply asking yourself which is the brighter of two things is something that you can't rely on. For instance here, the square that's labelled A and the square that's labelled B, which is the brighter of the two? Most people will have a clear thought as to which they think is the brighter of the two. But let me, without changing this image, let me just block off pixels to the left and let me block off pixels to the right. I haven't changed the squares A and B. Now I hope you're convinced that the grey of square A, which is the same as the grey on the left and the grey on the right, is exactly the same as the grey of square B. They are both the same as the grey masks on the left and the right. If I now take those masks away without changing A and B, now you know that those two squares are the same shade of grey. You know they are because I've just shown you they are. But your eye and your brain refuse to tell you that A and B are the same shade. You are now convinced that B is a brighter colour than A, even though they are not. Again, that is how easy the eye and the brain can be fooled by what you think you're looking at. If we were interested in scientifically analysing this, we would rely on the computer telling us the actual shade of grey or the actual colour of a pixel, not relying on our own perception to tell us what we think we're looking at.
Can I interrupt you, Steve?
Yep.
For me, B remained white and A remained dark grey. Is that just some aspect of some form of colour blindness for me? It's my particular eyes.
Are you saying that you see B as the same shade as A?
Yeah, when you move the remove the
Yep.
left and white, other than that diagonal strip, B still remained white for me and A remained dark grey.
Sorry, when the masks were there or when the masks were removed?
B is now still white.
Even though it's exactly the same shade as that?
It isn't. It isn't. It's much lighter.
No,
That's
it
what
is.
I'm saying. Is that my eyes then?
Well, okay. I can guarantee that that shade of grey that I'm pointing at is exactly the same as that shade of grey. There is no line between them. There is no change between them.
Right. Maybe other people see it as you say.
Compared to this, for instance, which is definitely lighter than that grey, and that's definitely lighter, the B is the same shade of grey.
For me, B and the slightly brighter white below it are more similar than B with the grey.
You're saying that shade compared to that shade?
Right.
These
yes,
two?
Those two are more similar than either of them with the grey.
These are very different. The grey on which B is sitting and that there is much, much brighter.
It must be my eyes, but it's a strange effect.
Can I ask, what screen are you looking at it on?
Just my normal laptop screen.
A laptop rather than...
The previous example you gave with the yellow and the blue, I agreed with you completely, or my eyes agreed completely, but not this one.
Is it the same here? Is that grey the same as the grey either side?
Yes, yes.
But that is the same grey as the one in which B is sitting?
No, they look totally different to me.
I can't see why that should be, because I checked myself to make sure they were the same. So they, unless the laptop is showing different intensities at different points on the screen, which is a possibility. It'd be interesting at the end of the talk to get feedback from other people to see whether or not they agree on that.
Yeah, apologies for interrupting.
It's okay. So this whole business of perception doesn't necessarily tell you what's actually there can be problematic. So for instance, we look at this image. This is an image from a colleague, and he says, I've been looking at strands of DNA on glass. So here we've got a few dozen strands of DNA, all of these little wiggly worms here, and the glass on which it's sitting is not, it is smooth, but smooth on a scale of microns. So if we were to look with a microscope, such as in this case an atomic force microscope, we have a resolution much less than the wavelength of light, and so the smooth glass background actually looks mottled. That's why we've got these grey mottling in the background. But that grey mottling doesn't stop us as humans looking at that saying, yes, I've got a grey mottled background, but I've clearly got a whole load of DNA which is brighter, and in this context bright means higher, so the DNA is sitting on the glass slide and is brighter because it's higher. So humans look at that and say, yeah, I can see that we've got a few dozen strands of DNA there. But if you were to try and get a computer to analyze that, it's a problem, because although we think that if we were to slice through that and ask what does the height as a function of position look like, it would be, well, we've got some sort of mottling in the background, and then every once in a while we've got something which is quite definitely higher than the background because it's whiter than the background, and we don't necessarily know what the scale is, but we quite obviously have objects that are sitting on this mottled background. We think that's what we're looking at. But actually, if we were to ask what is height as a function of position, it's much more like that. It is all over the place. In other words, the mottling of the background is actually almost as bright as the individual peaks of the DNA themselves. If we were to say, let's take a slice through this image, and it's actually that red slice there, we can ask, well, which of these peaks, which of these high points that appear to be brighter, which of those are because we happen to be passing through a strand of DNA, and which of them are just the background glass. And it's very difficult to look at that and get the computer to figure out which of those is DNA and which of those is just background glass. That red line there, we can see that it actually cuts through 3 strands of DNA, as indicated by the orange arrow there. And those 3 points correspond to that point, that one, and that one. So yes, the highest point there does correspond to a piece of DNA. But the second highest one doesn't, and the third highest one doesn't, and the DNA is actually those 3 green arrows there. How can we get a computer to recognize that's DNA, but everything else isn't? We, as humans, can pick out what's going on. Trying to program a computer to do the same job is actually quite complicated. It's one of these examples that humans find easy and computers find difficult. There are some problems the other way around, that there are some problems that humans find very difficult and computers find really easy, like counting lots of objects, that sort of thing. Counting 1 million objects is easy for a computer, difficult for a human. But this is the opposite way round. So when trying to analyze what's going on here, I first had to process the image in a way to make it absolutely clear which of these things are DNA and which of these things are the glass background. So I processed the image via an algorithm I'm not going to describe. I processed the image to turn the image on the left into the image on the right. And now the presence of an individual strand of DNA is a lot more black and white, metaphorically as well as literally. So now if it's black, it's a piece of DNA. If it's white, it's the background glass. And any small grey objects, I'm not too sure whether they're one or the other. But with an image on the right, it now makes it a lot easier for the computer to count how many strands are there and how long each strand is and how wiggly each strand is, for instance. But simply doing that is not easy, and it's an absolute prerequisite because it's so difficult for otherwise the computer to figure out what's actually going on in there. Let's have a brief look at some of the spin-offs along the way. So I wrote ImageSXM as a piece of software for carrying out image processing and image analysis, and they were mainly intended to be routines used in physics and chemistry to look at atoms and molecules. But the interdisciplinary nature of image analysis means that those same routines can actually be applied in other areas. For instance, I found that they could be applied in earth sciences and also in the medical sciences for looking at medical applications. So in the first case, I had a collaborator in Switzerland who was interested in analysing rock samples using polarized light. If you look at rock samples in a polarizing microscope, well, what do you see? Rock samples, if they are thin sections, are almost invisible. Most of the light goes through, but you can just about see individual grain boundaries where each grain corresponds to a particular type of rock or a rock with a particular orientation of its crystal structure. If you pass the light from the microscope through polarizing filters, either before or after or both of the rock sample itself, you can change the way the light behaves and you can get changes in intensity. If you're using circular polarizing filters, you can get changes in color as well. But the point is that by analyzing images like this, of looking at the intensity of light through rock samples passed through polarizing filters, it is possible to extract information about what must the orientation of the atoms in each of these little crystals, what must the orientation be in each of these crystals or crystallites in order to give me the variation in intensities that I see when I pass the light through polarizing filters and change the angles of the polarizers. So again, I'm not going to go into all the detail, but if images are obtained like this, black and white images obtained of different intensities that come through rock samples when passed through polarizing filters, It is possible then to analyze a whole stack of these images and extract information about the orientation of the crystals, because the orientation of the crystals as orientation relative to the polarizing filters changes the relative intensity of the light that's coming through. It's then possible to process those images to highlight where the grain boundaries are, and then produce images which are not supposed to be simply what would the coloured image look like in a microscope. These images are now coloured specifically about what is the orientation of the crystal in this particular crystal grain, and the colours here refer to the orientation as shown by this colour diagram. In which direction are the crystal axes of these crystals coloured according to whether they are pointing up or pointing sideways or pointing north, south, east, west, etc. So it's then possible for a geologist to start extracting information from images like this, knowing that each of these colours is telling them about the relative orientation of crystal axes, which then in turn tells the geologist about, well, what is the history of this rock that produced this particular set of orientations? Are they all in the same direction? Are they all in random directions? Can I deduce the chemical and physical structure of this rock, as well as the so-called texture of this rock, the collection and the distribution of crystal orientations, that tells me how this rock came to be and how it's been processed over millions or perhaps even billions of years. If any of you are genuinely interested in how image analysis is applied to rock samples in the earth sciences, well, my collaborator René Heilbronner and I produced this book a few years back, available at a very reasonable price from the authors. Medical spin-offs is what I've been spending more of my time dealing with. Over the years, I developed this suite of applications, which I called microscopy image analysis software for medical applications, or miasma for short. I carried out a number of projects. This is in collaboration with experts in other areas of the university or the School of Tropical Medicine, and I often got students to work on these as student projects to see how we could help our collaborators understand what it was they were looking at. So I've worked on, for instance, particulate matter, looking at carbon particulates in lung cells or lung tissues, looking at the development of parasites, looking at the circulation of blood cells. I'll come back to that particular application in a moment. Looking at features on the back of the retina, because that helps diagnose other problems with the patients. Looking at morphology of parasites, looking at the structure of cells, and looking at fibril structures as well as bacteria. In other words, looking at different morphologies as well as arrangements of these various biological structures. One of the latter projects I was working on was looking at human chromosomes. I thought this was a very interesting problem, especially one to set as a student project. It was a very ambitious student project because it's a very difficult problem. Here we have human chromosomes that have been dyed in order to show the banding patterns that are typical of the human chromosome set, or the karyotype. Here they are in cartoon form on the right-hand side. The 23 chromosomes, where the 23rd chromosome is either X or Y, depending on the gender of the individual. Each chromosome, you notice, has a unique banding pattern. And the question was, can you tell from images such as the one on the left whether or not all the chromosomes are present, and are all the chromosomes correct? In other words, do they have the right banding pattern? If the banding patterns are wrong, it indicates a chromosome abnormality. It turns out that that's very difficult to do with an image like that, not least because in this particular case you notice that some chromosomes actually overlap with each other, and therefore one chromosome is actually hiding the banding pattern from the chromosome beneath it. So that would mean you would need to take lots of different images from lots of different cells, all of which in principle have the same chromosome set, but are randomly oriented relative to each other, and therefore would hopefully not all have the same block of one chromosome hiding a chromosome beneath. But trying to correlate all that information, trying to tell from all of these perhaps hundreds of images, have we actually got all 23 chromosomes and are the banding patterns matching up correctly to indicate that there are no chromosome abnormalities? That is actually a very difficult problem, which is why at the moment the work is done in the Liverpool Women's Hospital by experts looking down microscopes at the chromosomes, and they are so expert at this they can glance at the image and they can say, yep, everything looks to be okay, and in a few minutes they can say, yep, everything's perfect except chromosome 13 has a problem. And they can do that in a matter of minutes, and then they can decide what are the consequences of that particular chromosomal abnormality. I thought rather naively that it might be possible to automate this task. It turns out to be a very, very difficult problem, but it was still a very interesting student project. Let's have a look at a couple of more complex systems. I'm going to pick out two that I think are worth having a look at in a little more detail. Microcirculation analysis, which is something I did about, what is it now, more than 15 years ago, and what I've been working on the last few years, investigating cancer. So first, microcirculation analysis. This was in collaboration with a consultant in Alderhay Hospital. He said some of our patients suffered from meningitis. It's basically a children's ward. If the children suffer from meningitis, it causes sepsis, previously known as blood poisoning. Sepsis means blood is not flowing around the capillary networks of the patient correctly. The microcirculation is simply a way of referring to circulation in the smallest of vessels, the capillary network. So it's not talking about how much blood is flowing through the main veins or arteries, it's how much is flowing through the capillary networks in tissue. And this was assessed by a small portable microscope which they placed under the tongue of a patient. So we're not really putting people under the microscope, we're actually putting the microscope under the tongues of people. The tongue is used because it's a way of getting very close to the capillary network. In principle, you could go through other areas like through the skin, but the skin is more likely to have fat and epidermis that gets in the way of actually imaging what's in the cell network of the microcirculation. So this is what it actually looks like. It's only about 1 millimeter or so on a side, and a video of about 10 or 20 seconds, I hope you can see, shows individual cells that are moving. In fact, that's all you can see in this image. You think you're looking at veins or arteries. Actually, you are only looking at blood cells. The only reason you seem to see shapes is because these veins or arteries are full of blood cells, and therefore they are chocker and seem to be solid. When there's rather lower flow, you can see individual cells moving. The veins and arteries themselves are not visible. So that's part of the problem. The clinician would like to know, is this circulation good or bad? In other words, are the cells moving at a reasonable speed through this capillary network? And that's rather difficult to work out. Again, a human can look at this and make a judgment call. You can look and say, well, over here they seem to be moving okay, but over there they don't seem to be moving very well. Over here the blood cells seem to be stopped completely, for instance, down here at the bottom. In other areas they seem to be moving at quite a healthy speed. An expert can look at this and make a judgment as to whether or not this patient has severe sepsis and whether their medication needs to be changed or not. And many times that's a fairly urgent decision. You can't leave this for a few weeks to decide. You have to decide in a few hours whether or not this patient needs to have their medication changed. So the question is, rather than having experts spend their valuable time analysing hours and hours and hours of video, is it possible to automate this task? So again, I thought, well, yes, it should be possible. But part of the problem is, with no disrespect to the individuals taking the data, the quality of the data is such that it's very difficult to see individual blood cells. If we blow that square up and say what's actually going on inside that blood vessel, for instance, what's going on in this particular blood vessel there, well, yeah, you've got a few pixels, some of which are darker than others. But how are you going to tell which of those are blood cells, and how are you going to track their movement? Especially when you bear in mind that the blood vessels themselves are not visible. This particular technique only images the cells, the hemoglobin, which absorbs a particular wavelength of light, and so the cells are imaged. It's only the motion of the cells that gives us the impression that we're seeing blood vessels. So how do you identify and then ideally quantify the motion of a blood cell relative to an invisible vessel in a sequence of video images like the ones you've just seen? Especially when you bear in mind that we can't simply watch what happens to one cell, because the entire network of vessels could be moving. Have you ever tried to get a young kid to sit still while you put a microscope under their tongue? No. Any attempt at quantification of the motion of cells would have to deal with the fact that the video is imperfect. There will be lots of reasons why the cells might appear to move. For instance, the entire view might translate because you can't fix the microscope to the bottom of the tongue. The tongue is a muscle, and you're placing the microscope under the tongue. So it might be that over the course of 10 or 20 seconds, the whole field of view moves. It is also possible that the magnification changes, because the magnification, this blood vessel, is just below the surface. Depending on the pressure with which the microscope is placed onto the tongue, it can change the apparent magnification. So the vessel network may appear to get larger or smaller. It's also possible, because you can't hold it perfectly still, that there might be a rotation involved as well. And the tongue is a muscle, so there will be distortions, because you can't possibly hold your tongue completely still, and you don't want to anaesthetize the patient, because that would change the circulation of the blood. In fact, all of those will apply. Changes to translation, rotation, distortion, etc. And then on top of that, the patient might be on a ventilator, and there will be a heartbeat changing the speed at which blood flows through the capillary. So with all of these problems, it is clearly a major task to figure out how to get a computer to figure out what an expert can do in a few minutes. I had to apply a number of different techniques. Again, I'm not going to go into the mathematics, but there are different techniques that can be applied called correlation techniques that allow you to see when objects are moving and to be able to take into account the distortion of the network due to translation or rotation or distortion. And then it was possible to say, right, relative to the invisible network, how fast are these blood cells actually moving? And after what seemed like years and was probably many months of trying to code this, I ended up with this sort of thing. If we take a patient which is relatively healthy, in other words, a control patient, and we ask how fast are the blood cells moving, at a given speed, how many vessels appear to have a motion of blood cells at that particular speed? We find that for a healthy patient, most of the blood seems to be moving through the vessel network at a speed of about 500 microns per second, or about half a millimeter per second. One of the first things I did was ask the clinician, my software appears to give me an answer of 500 microns per second for a healthy patient. Is that right? And the clinician said, don't know. I can tell you when it looks right, but I can't actually tell you. I'm not sure anybody has actually calculated how fast blood cells move through a capillary network for a healthy human. I found that rather weird, that as a physicist, I was trying to calculate numbers. I wanted to make sure I was calculating the right numbers, but I didn't have an absolute standard to work by. Experts know when things are going well and when things are going wrong, but that doesn't mean that they quantify it in the same way that a physicist would look at it and say, I need to work out how fast those blood cells are moving. What is important, that the difference between a healthy individual and an individual with sepsis was visible. In other words, you can see that for a patient with sepsis, it is still possible that some of the blood cells are moving through the capillary network at reasonably high speed. But rather unfortunately, quite a lot are moving at rather slow speed, slower than you would expect for a healthy individual. And it's that difference that allows us to tell the difference between a patient in which the blood flow looks fine and one in which there is a problem. Now, I didn't believe that writing software that would analyze the motion of cells and then spits out histograms, I figured that wasn't useful. So I figured it would be more useful to actually color a map of the vessel network according to whether the speed looks reasonably healthy, i.e. greenish colors, speeds of 600 or 800 microns a second, or for a sepsis patient, when we look at the color map, we see that there are rather a lot of reds and oranges corresponding to speeds well down here in the speed color map on the right hand side, the legend there. So I figured that producing colour maps would be far more useful if we ever wanted to get this into the NHS. What we really want is a piece of software that can show visually on a screen whether or not everything looks green, i.e. okay, or whether there's an awful lot of red, in which case a consultant needs to be called to make sure that they can decide what to do with this patient because they are suffering from severe sepsis. So that was a very interesting challenge because it was such a difficult problem to deal with, and producing these colour maps seemed to be the right way to go. My collaboration with the clinician, the consultant in Alderhay, came to an end because ultimately he retired, and it would be nice to hope that somebody will pick up on this idea and perhaps see it through to completion. Let me finish with what I've been doing more recently with investigating cancer. And I apologise now if I say I mean we, because I work within a group of physicists working in the University of Liverpool, and we've been working in this area for quite a few years now. So it describes research that has been funded through a number of different research grants, Engineering and Physical Sciences Research Council, through Cancer Research UK, a charity, and through the National Institute of Health and Care Research, NIHR. It was started a few years ago to try and answer the question, can we identify an infrared absorption signature for tissue that is likely to become cancerous? In other words, if we shine infrared light on tissue, certain wavelengths of infrared light will be absorbed according to which molecules are present. Can we use that as a signature to tell us this tissue is likely to become cancerous in the future? In other words, we can reduce that down to the slightly pithier question, can we detect cancer before it is cancer? We started by taking infrared light from an accelerator. I'm not going to talk through all the details, but there's an accelerator at Daresbury Laboratory. It was originally called ALICE, and part of that accelerator was designed to produce infrared light, and it had the sort of frequencies that we were interested in using. How does it do that? Well, again, we're not going to get bogged down in the details, but basically if you take an accelerator particle, in this case an electron, and you get it to wiggle through a series of magnets as shown here, then the light produced by that electron that's getting tugged backwards and forwards, that light will have a frequency or a wavelength that depends on the array of magnets that you're using. So Alice had an array of magnets that was suitable for producing infrared light. And we used that infrared light to get an idea of how cells absorb the light. Here we've got a few images. It's not so much the morphology of the cell we were interested in. It happens to be a cell associated with esophageal cancer. But we were more interested in not the morphology of the cell, but how the cell absorbed different wavelengths of infrared light. And we hoped that there were particular wavelengths that told us about the nature and the chemistry and the biology of the cell that gave us a clue as to whether or not the cancer would develop in the future. We've now changed from using accelerators at Daresbury Laboratory to using infrared sources of light in the lab. Again, I'm not going to go through all the details, but I'll answer questions on that later if you're interested. We now take images of samples in which each pixel is not simply a number, each pixel is an entire infrared spectrum. In other words, it's the absorption of infrared light at lots and lots and lots of different wavelengths, or wave numbers as we call them. Think of this either as the energy of the infrared light or the wavelength of the infrared light. It doesn't really matter. There is a particular pattern produced by essentially every molecule, and in this case there's a tissue which produces these characteristic peaks. The question is there any sign of any changes in this spectrum that tell us that not only are these regions of the tissue perhaps a different type of tissue, does it tell us whether healthy tissue is different from tissue which is going to become cancerous? Can we use the absorption at different wavelengths to identify different types of tissue, including a particular type of tissue that we know is a signature that is going to become a problem, going to become cancerous in the future? Infrared has the disadvantage that it's a longer wavelength than optical wavelengths, and so generally the amount of detail you can see is rather less than the amount of detail you can see in, for instance, an optical image, as shown on the left-hand side. One of our PhD students developed a technique in which the high-resolution optical information could be combined with the lower resolution infrared, which contains chemical and biological information about how the system is behaving. Those two images could be fused together, not surprisingly called image fusion, such that we can maintain the high resolution of the optical image as well as the chemical information telling us what is going on with the biology in these various parts of the sample. So what type of samples are we dealing with? What types of tissue have we been studying? Well, we've spent a fair bit of time looking at epithelium tissue. That covers essentially all surfaces of the human body. That doesn't just mean skin, it means surfaces such as, for instance, the esophagus or inside your mouth. We've studied esophageal cancer cells in the past, but we've spent quite a few years now focusing in detail on what's happening with oral epithelial dysplasia. This is a change in the cells in the lining of the mouth that might be benign, but they might be an indication of early signs of cancer. The question is, can we study OED and see whether or not we can pick out the signs that these are going to become a problem? We want to improve the accuracy of prognosis, so that if you find an abnormality in the mouth, let's say a little white patch, a little lesion, can we tell if that is not going to become a problem or whether it is likely to become a problem in the future? And then we can make sure that the individual, the patient, gets seen as often as possible to catch cancer as early as possible. We talk about whether or not the lesion will transform into cancer or whether it won't transform into cancer. So the problem we're dealing with is transforming or non-transforming. So here, for instance, I've got a picture of two lesions. The left-hand side, there's a lesion, a little white patch on the gum of the cheek, the inside cheek of this particular patient. And here's another white patch on the underside of the tongue of a patient. Now, in this particular case, one of these lesions is going to transform into cancer and the other isn't. How do we know? Because these are historical samples and we've been able to follow the history of the patients. These were images taken a few years ago. But the question we're trying to ask is it possible to tell by analysing these white patches, analysing these lesions, is it possible to tell which one will transform into cancer and which one won't? We think the answer is in the infrared spectra. The catch being the infrared spectra look very, very similar. There are tiny differences in there, but again we come back to the fact that humans are not very good at seeing tiny differences. We tend to concentrate on the big picture. And if we were to look at those two spectra, a human would say, well, they're the same. They've got all the peaks and troughs and bumps, etc. in the same position. But by taking spectra such as these, and not 2 spectra, I mean we take thousands or hundreds of thousands or millions of spectra, and then we start to see tiny differences. But we don't see them by eye. We have to resort to writing computer code that looks specifically for tiny differences in spectra that might be indicative of whether or not this is going to transform into cancer or not transform into cancer. So one of my PhD students, who's now a postdoc, is an expert in writing code that will pull out these tiny differences to tell us what's actually going to happen, or at least predict with as much certainty as possible what is going to happen. So again, no time to talk about all the details, and I'm not going to show you thousands of lines of code as to how this is done. But the problem can be summed up as it's a sort of artificial intelligence. We are trying to train a computer to recognize the difference. Here's a load of spectra that correspond to a lesion that did transform into cancer. Here's a load of spectra from a lesion that didn't transform into cancer. Can we get the computer to recognize the difference? In principle, it's no different from any other AI problem where you're trying to train it to tell the difference of different things. For instance, what if we wanted to train AI to tell the difference between a horse, an octopus, and a dog? Now, humans have no problem with this. Again, it's easy for us. This is something we can easily do. It's actually quite tricky for any other piece of computer software to do it. How do we know that's a horse? Well, because we've seen thousands of horses and we know what they look like. How do we know that's an octopus? Well, it might be the colour, it might be the fact that it's got 8 legs. Bear in mind, we can't see 8 legs in that photograph, but we can still work out it's an octopus. And dogs, well, dogs come in dozens, no, hundreds of different breeds, but we usually have no problem whatsoever in recognising a dog when we see one, even if it's got a flower in its mouth. So how do we train AI to recognise the difference between these 3 animals? Well, it's not enough to show it one picture of each. We have to show it hundreds, thousands, tens of thousands of images of horses of different colours, of dogs of different breeds, of octopuses of different colours, from different angles, on different backgrounds. Only when we've trained it on lots and lots and lots and lots of possibilities can we hope that the AI actually learns to recognize the difference between a horse, an octopus, and a dog. When we think we've got it right, if we've tried to train a bit of software to recognize the difference, we can't just assume that it then works. We have to validate it. In other words, when we've got what we think is a bit of software that has been trained to recognize the difference, we check by saying, right, let's take a picture of a horse that it has never seen before. This is not one of the set that we've used to train it on. This is a picture of a horse that we know it hasn't ever seen before. Does it then recognize it as a horse or an octopus or a dog? We give it that picture, it's never seen it before, and it correctly identifies it as an octopus. We give it a picture of a horse it's never seen before, and it correctly identifies it as a horse. But every once in a while, it gets it wrong. We give it a picture, and it says, that's an octopus. Now, again, humans would instantly say that's wrong. But the AI doesn't know that. The AI has taken the best guess it can, and it's come to the conclusion that that's a dog. Correction, that's an octopus, not a dog. Why did it come to that conclusion? That's part of the problem with AI. With some AI, you can't tell why it came to that decision. Maybe it thought that dog was an octopus because, well, how many legs has it got? Well, you can't really see how many legs it's got. And you certainly can't count 8, but you don't often count 8 on an octopus anyway. Maybe it's the fact that the dog is splashing around in water, and most of the pictures of octopus have water somewhere in the background. Not always, but sometimes. If your AI is written in such a way that you can't ask it, why did you come to that decision? Then AI might be useful, but it is of limited usefulness if you can't ask it why it came to that decision. So if it gets it wrong, we really need to know why it gets it wrong. So the software that's been written within the group in physics at the University of Liverpool, we've paid particular care not only to try and get the best software we can, write the best software we can to give us the best outputs, we've also written it in such a way that we can test and validate how well it is doing. So if it gets it wrong a few % of the time, we need to know it gets it wrong a few % of the time. And ultimately, over time, we might be able to improve, if we know why it's getting it wrong, improve the algorithms, improve the software, and do a better job of predicting when tissue is going to become cancerous. So we don't tend to call what we're doing AI, but it's based on the same idea of train software to recognize something, in this case spectra rather than images of horses. We're training it on spectra that have particular infrared signatures in them, and it's picking out those differences, those small differences, to tell us what's going to happen to that tissue. The group I'm working in is called SciScan, Spectrochemical Infrared Analysis Scanning for Cancer. We're developing a device, a medical device, which we hope one day will end up in the hands of surgeons, such that they will be able to take this laser-based device with infrared lasers and will be able to, for instance, whilst they're excising perhaps a tumour from a patient, they'll be able to wave the wand over the tissue that's been left, and they'll be able to tell, because the wand will go beep or flash red or flash green, and will be able to tell the surgeon, yep, the tissue that you've left behind is not going to transform to cancer. In other words, you've caught all of it. If the surgeon leaves behind a little tissue which is still likely to become cancerous, then they need to resect a little more before they close up the patient. So we're intending to produce medical devices which can be used in histopathology laboratories for screening biopsy samples, but one day it would be nice to think that what we're doing might end up in the hands of surgeons. A reminder that science is not a solitary affair, so I've been talking to you about what I have been doing over the years, starting with atoms, working through molecules, spinning off into the earth sciences and now into the medical sciences. But all science is collaboration. So I'd like to acknowledge all of my collaborators, fellow staff, students over the years, staff and students in other departments in the university and in other universities around the world. And I'd like to acknowledge the funding bodies as shown in the bottom right hand corner for supporting my research over many decades. Thank you all very much.